Hi everybody, my name is Alex Javrankov. I'm the founder and CEO of In Silico Medicine, and welcome to the PHARMA.AI Day, where we cover Biology42, PandaOmics, and DORA, our flagship platforms for target exploration and scientific discovery. PandaOmics has been on the market for now close to 5 years. This is going to be our 5th year in operation. So it already acquired very substantial level of validation, both utilizing our own internal drug discovery programs. So now we've nominated 20 preclinical candidates, 10 of the drugs reached human clinical stage, and 2 went into phase 2, 1 completed phase 2A with safety and unexpected efficacy. We also have massive validation from the many customers who are utilizing these tools, and we continuously integrate massive amounts of feedback. So every time you use it and provide feedback, we are improving the tool. So please do provide us with more feedback after you try the tool, even if you don't like it, especially if you don't like it. PandaOmics has acquired superpowers, so now you can onboard and offboard onto and off the system automatically. You can utilizing it from the comfort of your home with a credit card, and you can select how long do you want to use this platform for. It also became much more flexible in terms of the use of your own data and connection to many other systems and platforms that you might have for target sciences.
Hello everyone, and thank you for joining us today. We're excited to share the latest updates to PandaOmics, an integral part of the PHARMA.AI suite. Here is a brief overview of the novel functionality we'll be reviewing today. This year, we've significantly expanded our data warehouse. We've doubled the list of tracked clinical trials by indexing international clinical trials databases, ensuring that you can access the latest insights from ongoing research. We've also updated our drugs and trials database, which links clinical trials to investigational drugs. In many cases, we found new information about the drugs from press releases, papers, and reports, information that is unavailable in the public clinical trials databases. Next, our knowledge graph visually links genes, diseases, drugs, and processes by extracting this evidence from clinical trials, papers, and grants. With the improvements in scientific reasoning, we were able to double the number of extracted links. Finally, our annotation team created meta-analyses for over 1,000 diseases. For each disease, they manually triaged omics datasets and configured the experiments, collecting reliable insights. You can find these meta-analyses right on the corresponding disease pages in PandaOmics. Our data warehouse is also available for purchase separately from PandaOmics. It can be deployed as a SQL database on-premise or in the cloud, and regular updates make the most recent information quickly available to you. If you are interested in accessing this data, please contact us at PandaOmics@insilico.com. The Knowledge Graph is an interactive network that integrates various biological entities, including genes, diseases, chemical compounds, and biological processes. Each entity is linked by distinct edge types, such as activation or inhibition. In this latest PandaOmics release, we've re-engineered the Knowledge Graph engine by introducing new relationship types, including biomarker, target, and therapeutic connections, to better serve applications in pharmaceutical research and clinical translation. As I explore the Knowledge Graph, I can see how biological pathways such as apoptosis and cell cycle regulation are interconnected with key genes like MET, TP53, and EGFR. If I want to delve into specific relationships, say between the cell cycle phase and the EGFR gene, I can simply click on the corresponding link. This opens up supporting citations for that connection, allowing me to quickly assess the depth of research available. From there, I can click on a citation to view a summary of the relevant paper, enabling a quick impression of its relevance before deciding to read further. Additionally, genes and diseases are highlighted within the abstract and linked directly to the dedicated PandaOmics pages, making it easy to explore each element in more detail. I also have the option to talk to ChatPanda GPT, an interactive AI bot that's aware of general biological data and has been augmented with specific insights from the knowledge graph. ChatPanda GPT offers immediate information on therapeutic targets, compounds, and more. I can interact with ChatPanda GPT either by selecting one of the predefined questions or by formulating custom queries based on my research needs. For example, if I'm interested in identifying genes associated with lung cancer in patients who have never smoked, I can pose this question directly. ChatPanda GPT will then provide information on relevant genes, highlighting those with notable genetic alterations, such as EGFR and ALK, which are particularly relevant in this patient subset. Let's explore how we can identify drug targets. Recently, we introduced an intuitive mechanism for the automatic creation of meta-analyses, enabling users to formulate queries in plain language instead of manually assembling datasets. To start, I'll click on the Create New Meta-Analysis button in the Data Manager. Here, I can enter my query, for example, lung cancer in never smokers compared to smokers. PandaOmics performs an AI-enhanced search and presents a list of datasets relevant to my query. Each dataset includes key properties such as sample size, tissue type, and technology, allowing me to assess its suitability for inclusion in the meta-analysis. Moreover, I can verify if each dataset has passed manual quality control and examine dataset design to ensure it aligns with my research focus. If I determine that a dataset doesn't fully match my needs, I can exclude it by toggling it off. Once I've finalized my selection, I click on Create Comparisons button, and PandaOmics will try to automatically generate comparisons based on the chosen datasets. While this process may take some time depending on the dataset volume, I can navigate away from the page without losing progress. Once all comparisons are generated, I can inspect them in detail and review information, such as the number of cases and controls and the fraction of differentially expressed genes. If a comparison doesn't meet my criteria, I can easily exclude it from further analysis. When I'm satisfied with the final list of comparisons, I proceed by clicking the Run Meta-Analysis button and specify the therapeutic area as non-small cell lung carcinoma. This initiates the scoring process for the meta-analysis, which may take some time depending on the number of comparisons included. Once complete, we'll have a prioritized list of potential drug targets. Once the score calculations are complete, I can access the meta-analysis results in the Data Manager. In the Target ID tab, I can adjust a range of parameters to refine the final ranking of potential drug targets. So, what's next? It's simple. Sign up today to expand your horizons of omics data analysis and visualization.